Oxidative phosphorylation. Oxidative phosphorylation occurs in the cristae, inner mitochondrial membrane, but also involves the matrix and intermembrane space. It involves the electron transport chain, ETC, movement of protons across the inner mitochondrial membranes, the chemiosmotic theory, and the production of ADP via ADP synthase. Most ATP is made in this stage. It requires NADH, FADH2, and oxygen. The electron transport chain is a collection of proteins found within the membrane. 1. Hydrogen atoms collected by coenzymes NAD and FAD are delivered to the electron transport chain present in the membranes of the cristae of the mitochondria. 2. NADH and FADH2 are both oxidized. NAD is oxidized by NADH dehydrogenase, which is the first protein in the electron transport chain. These hydrogen atoms dissociate into hydrogen ions, H+, and electrons, E. 3. Electrons enter the ETC. Energy is released during redox reactions as the electrons reduce, then oxidize the electron carriers as they move down the chain. This produces energy for H+ ions, protons, to be pumped out of the matrix across the membrane, out into the intermembrane space. This creates AH+, or proton gradient. 4. H+ ions then diffuse back through the matrix from an area of high concentration to an area of low concentration. However, due to the impermeability of the membrane to H+ ions, they diffuse through ATP synthase. This results in the synthesis of ATP from ADP and pi. 5. At the end of the ETC, electrons combine with hydrogen ions and oxygen to form water. Oxygen is referred to as the final electron acceptor. The ETC cannot operate without oxygen, so this process is aerobic. It is considered oxidative because electrons are lost and oxygen is the final electron acceptor. Oxidative phosphorylation is not a completely efficient process. Some H+ ions leak back into the matrix. Oxidative phosphorylation, in particular, steps 3 and 4, is an example of the chemiosmotic theory, a theory in which it is proposed that the ETC is arranged such that it generates an energy-rich proton gradient across the inner membrane of a mitochondrion. The energy is then used to drive the phosphorylation of ADP through a membrane-bound ATPase, ATP synthase. The inner mitochondrial membrane has several properties that allow chemiosmosis to occur. It is mostly impermeable to H+ ions/protons. It has a large surface area. It contains ATP synthase, stalked particles. The intermembrane space becomes more positively charged and its pH decreases as a result of oxidative phosphorylation.